Viruses: history, structure, growth cycles, diagnosis, and disease. These are all items that we will discuss regarding viruses. Why do these tulips look this way? The two different colors and all the little stripes. Well, some of you have already determined it's due to viruses. Viruses have caused the insertions within genes that are responsible for colors in the tulips, and actually makes them highly desirable as flowering plants. Tulips still are bought and sold very much like other kinds of commodities, and in the 1700s, because of the wild diversity of colors and shapes, tulips became worth their weight in gold, practically, because of their beauty and the various types of colors and the effect that these viruses had. Of course, they didn't know at the time these were viruses that were causing the color changes, but these dramatic differential colors and the stripes are all highly desirable and aesthetically pleasing characteristics of these types of virus-infected tulips. Viruses are highly debated, or maybe even hotly debated, amongst scientific circles because viruses challenge cell theory. There are some biologists who are classically trained who would not even imagine or think of using the word organism when describing a virus, because viruses are acellular. And in particular, in classically trained biology, with cell theory, all organisms are made of cells. Well, viruses do not have cell structure, so oftentimes they're described as neither living nor dead, because when they're not inside of a cell replicating and forming new viral particles, they're essentially just what we would now call in microbiology microorganisms in search of a host. Now, in microbiology, most of us have no problem at all describing viruses as microorganisms. They just have very unique, highly organized, and very specific types of growth or life cycles. That doesn't mean that they fall underneath the definition of being an organism with cells. They are still acellular in their most fundamental shape. But part of the life cycle of a virus includes growth inside an unwilling host cell, whether it would be prokaryotic or eukaryotic. There's virtually a virus that can infect every kind of cell. Viruses tend to be very species specific. So the virus that causes leukemia in cats will not cause leukemia in people, at least that we know of so far. Viruses must replicate inside of a host cell. And again, they're very specific for what kind of cell they can grow in. Viruses, once inside of a cell, will take over all of the, or at least most of, the normal function of the cell. Many times it is all of the function. And direct all of the metabolic processes, biosynthesis, typical types of activities that are occurring inside of a normal growing cell, and direct all of the function of that cell to the manufacturing of viral components. The cell becomes a slave to the virus. And that's one of the reasons viral infections are difficult to treat. The virus is essentially hiding inside of a cell. So viral infections cannot be treated with antibiotics from the traditional definition of antibiotic. Traditionally, an antibiotic is a chemical produced by a fungi to kill a bacterium. And of course, viruses are neither bacteria nor cells, and so you really need an antiviral. We need specific drugs to treat the viruses, to inhibit or destroy them. And of course, the difficulty is getting the drug inside of the cell that either doesn't harm the cell any further and impedes or slows or prevents the virus from growing, or perhaps just destroys the cell that's infected completely, which is essentially what your immune system will do. If your immune system recognizes a cell that's infected with a virus, that virus has essentially doomed that infected cell to be destroyed. Because your immune system has both chemical and cellular factors in the form of white blood cells that are constantly patrolling and looking for virally infected cells, or cells in your body that are different and different enough to be eliminated. So at this time, we don't have any way to cure a cell that's infected with a virus. The only way to get rid of it is to either prevent the virus from attaching and gaining entry, so it has nowhere to go, and thus we stop its growth, or we destroy the cell that has already become infected. Now, in terms of history, there are just a few individuals that we need to learn about. One is Adolf Meyer. In 1886, which was part of the golden era of microbiology, where all of the numerous kinds of disease organisms and various characteristics of microorganisms in terms of bacteria were being discovered and recorded and reported. Adolf Meyer was the first individual to notice that tobacco mosaic disease, which would affect a commercially important crop like tobacco, was contagious. In other words, easily transferred from one plant to another. With a contagious disease in this type of commercially important plant, like tobacco, it could be devastating to a farmer who was relying on that crop for their livelihood. Meyer noticed that if you took a leaf and noticed that some of these leaves in these photos have either mottled yellow patches, which is a little bit evident in the left-hand picture in the lower left, or even some brownish dead patches along with some of the yellowing on the leaves, those are all signs or symptoms of tobacco mosaic disease. If Meyer took one of the leaves with these yellow patches, especially with the brown dead-looking patches, and rubbed them onto a healthy tobacco leaf, within a short period of time, that healthy tobacco leaf would now show signs and symptoms of TMD, or tobacco mosaic disease. This is the first time anyone had recorded that this type of disease was transmissible and contagious. In other words, transferred from one plant to another, and it can be easily transferred, and that's why we're using the word contagious. Later on, we're going to have contagious as a testable term when we go through immunity and host microbe immune responses, but for now, we're just using it as a descriptive to tell us how easily whatever this disease was and what it was caused by could be easily transferred from one tobacco plant to another. An unusual and new finding that no one had ever noticed before. Dmitry Yawanowski in 1892, a few years after Meyer noticed or was able to report that TMD was contagious, determined that whatever the cause of TMD was, it was something filterable. In other words, smaller than a bacterium. He used a specialized device called a Chamberlain candle filter. The Chamberlain candle filter was a device at the time that was the most powerful filtering device that they had available. It would filter out debris. If you took the leaves of the tobacco plant, ground them up in some liquid or some solvent, and then filtered out all of the liquid, passed the liquid through this Chamberlain candle filter, this filter was capable of essentially capturing all of the tiny little bits and pieces, including bacteria, and remove it from that liquid. Well, when they took the liquid, it was still found that whatever was in that liquid was still causing TMD. And so he was the first person to demonstrate that TMD was caused by an acellular infectious agent. Now, at the time, the word virus meant poison. In fact, for hundreds of years, the word virus had been used in terms of poison. They really didn't realize that these were actually microorganisms or some type of entity that could essentially interfere with normal cell function. So he knew it was something filterable that was causing TMD. We have a few more folks who contributed additional information that gets us to the point where we now know that TMD is, in fact, a microorganism-caused disease. Martinus Weidrinick was, in 1895, the first person to suggest that TMD was caused by an infectious agent, that the fluid that could be extracted from crushed leaves was a contagium. Now, we clearly have the concept now when we look back in history at the contributions of Adolf Meyer, Dmitry Iwanowski, that they were working with some kind of contagious organism. But really, it was Weidrinick that was the first person to sort of galvanize these concepts. Even Iwanowski didn't call the material a virus. He did think there was something maybe infectious in his fluid. And Weidrinick consolidated those concepts and those ideas, truly using the word virus probably for the first time in the context of an infectious agent instead of a poison. So instead of looking for some kind of chemical that could cause TMD, Iwanowski got them down the right path, and Weidrinick thoroughly confirmed the idea that it was caused by a microorganism, potentially, or some kind of infectious agent. In 1935, Wendell Stanley, who was a virologist now working with TMV, tobacco mosaic virus, was the first scientist to crystallize TMV and examine it using X-ray crystallography and also using a transmission electron microscope. He really was the first person to allow us to get a glimpse of what viruses really looked like, and you can see in the photo on the left, they simply look like little rod-shaped structures. When you could zero in and look really closely at the magnified image on the right-hand side of that photo, you can just barely make out that there is some type of surface structure on TMV. Almost looks as if you have stacks of plates, one on top of another, that form these long rods. Well, in 1946, Stanley did win the Nobel Prize for his contribution for the isolation, crystallization, and identification thoroughly of TMV. Now, oftentimes we'll use the word virus. Occasionally we can use the word viral particle or virion. Sometimes scientists reserve the word viral particle or virion for a newly replicated or a newly produced virus. Other times we simply use those terms interchangeably. But we're still talking about the same entity. We're still talking about a virus. And scientists who specialize in the study of viruses are called virologists. And it's an important area. Lots of activity and lots of research are occurring even today to study and learn about all the new kinds of viruses and even some of the old ones that have been around for a long, long time. There are a variety of ways that viruses are classified or grouped together. One way is to simply look at the outer covering. There are two categories of viruses based on their outer covering. The first are the naked viruses. Naked viruses are the most fundamental in structure, if we're going to call it a virus. We have a nucleic acid core. And for viruses, that's only going to be DNA or RNA. We are never going to have both packaged inside of the outer covering that we call a capsid in a virus. So only DNA or RNA. We have a capsid as the outer covering. That's the entire complete structure. The capsid is composed or made up of capsomeres. So notice that the capsomeres on this little illustration are the little blue spheres. When we assemble a sufficient number of the little blue spheres, we create the outer blue covering that's called the capsid. Now also notice there's a shape. This is a polyhedral virus. This is a polyhedral naked virus. We can often classify and group viruses together based on shape. And so we have polyhedral viruses, bullet-shaped viruses, rod-shaped viruses, viruses that are categorized or classified based on their shape and their outer covering. So we have this polyhedral naked virus. We could have polyhedral bullet-shaped viruses. Starting to zero and narrow it down based on their shape and their outer covering. Instead of being a naked virus, some viruses have an envelope as their outermost covering. Now we'll see as we go through the slides and discussions about viruses, the viral envelope is actually a piece of the host cell's cytoplasmic membrane. So at one point in time, this envelope, which is actually a construct of phospholipid, came from the cell in which this virus had been replicating and growing. So we have a viral envelope. Notice that this one has spikes sticking out of the surface. Spikes are not exclusive to envelope viruses. We can have naked viruses that have spikes. Just so happens that we don't have an illustration here right this second that has a naked virus with spikes. But we have this outer envelope. On the inside, we have a capsid that's surrounding a nucleic acid. So you can think of essentially that envelope viruses have an additional outer covering. It's as if we take a naked virus and add another phospholipid outer covering to give it a little extra protection. Also notice that based on shape of the capsid, this is being described as a helical virus. You can't really see the rest of the picture because of the cutaway, and it's hard for artists to depict all of the parts of many viruses because they can have very geometric designs and very interesting arrangements of their structure. But this is described as an enveloped helical virus, an example of another way or another group in which viruses can be classified. When viruses undergo replication, or what we might consider as its life cycle, the first thing every virus must do in order to produce new viral particles is to attach to essentially an uninfected host cell. It has to attach. The next step that has to occur is penetration of the nucleic acid. Now, penetration of the nucleic acid could simply mean inserting the nucleic acid into the host cell, almost like injecting it with a syringe and needle, or perhaps the whole entire viral particle is brought inside of the host cell, and it's once inside that the uncovering and the revelation or the release of the viral nucleic acid occurs. After penetration or the introduction of the viral nucleic acid, the viral nucleic acids are fairly powerful in their ability to essentially take over many of the normal functions or redirect many of the normal functions of the host cell. And now this cell will start to produce viral proteins and viral nucleic acids. So that biosynthesis is no longer for cellular needs, but instead the cell has become a slave to the virus and has begun to produce viral proteins and viral nucleic acids. As these proteins and nucleic acids are synthesized, bits and pieces are assembled into larger, more useful pieces until eventually an entire capsid has been constructed with a complete, ideally unique and identical copy of the genome of the virus packaged inside. That's described as maturation, when we have a complete copy of the original virus completely sealed and packaged inside of a newly formed capsid. Once we have this fully functional, fully formed virus or viral particle, now we have to get it out of this infected cell. There are various ways that we have viral release occurring. If we're looking at both prokaryotic and eukaryotic cells, there are many viruses that simply bust out of the infected cell. This is called cell lysis, L-Y-S-I-S, and just as a very common means of viral release from an infected cell for both prokaryotic and eukaryotic examples. Now for animal cells, we do have an alternative to cell lysis, which is budding. That simply means the virus is emerging from the cell sort of bit by bit in little blips or blobs. And we do have some illustrations trying to show you how the little viruses are being released slowly in these little packages from the virally infected cells. And that's called budding. Alternatives to lytic release or budding would be lysogeny for prokaryotic cells and latent infections for eukaryotic cells. We'll have examples of both in a few more slides. Here's an example of lytic bacteriophage infection of a host cell. Bacteriophage. That's another word for bacterial virus. Phage refers to mouth or eating. And decades ago, when scientists were working with bacteria, especially, let's say, samples of bacteria from lakes or streams or ponds, they noticed that occasionally they would have a bacterial culture that would appear to be eaten up by some mysterious little invisible entity that was much smaller than the bacteria. Well, these are bacterial viruses. And the bacterial viruses have a way of gaining entry into the bacterial cell. But in order to get out, we're going to see in the next slide, we're going to have to bust out of this bacterial cell because of the cell wall that prevents us from emerging any other way. So to get started, remember, every good virus has to attach. And on our exam, when we test on information about viruses, please be sure you know the stages and what happens at every stage in a typical cell cycle or replication cycle for viruses. Here we have, in this illustration, a bacteriophage that looks very much like a little alien that might be used in a science fiction movie. Really typical kind of traditional bacterial virus. Notice at the top we have the capsid, in which, in this example, we have viral DNA tangled up and packaged very tightly inside the capsid. The rod-shaped assembly that we see in the picture, this labeled a sheath, actually is very much like a syringe. And then at the very tip end, at the bottom, where this bacteriophage is attached to the cell wall of the bacterium, we have almost a needle-like structure that's able to penetrate through the peptidoglycan layer, through any outer membrane, peptidoglycan construct, and even through the cytoplasmic membrane. So the sheath, along with the injection or the transport tube, act very much like a syringe and a needle. Now notice that we have what are described as tail fibers. These are little tiny structures that allow the virus to attach firmly to the outer covering of this bacterium. Once we have attachment, in other words, the tail fibers anchor onto the surface of the bacterial cell, the sheath will contract or squeeze together, very much like a little spring, or as if you were pushing the plunger down on a syringe. And notice in the bottom illustration, this allows the virus to inject its genetic material. And what's the term we use for the injection of the viral genome into the host cell? Penetration. So the phage penetrates the host cell, injects its DNA, and now this viral DNA is going to redirect much of the metabolic processes inside of this host cell to essentially synthesize viral components of proteins and nucleic acids used only for the virus. Biosynthesis is the next step, with the production of viral components, nucleic acids, and proteins, as directed by the phage DNA that was injected into this host bacterial cell. Once the components are fully assembled into new viral particles, those new viral particles are released by cell lysis, and now each one of those could go on and attach and continue another cycle of replication or production of new viral particles, just like the first time or this first illustration here. Here we have an example of an alternative. Many viruses cause lytic infection.
They could go through the typical life cycle of a lytic virus with attachment, penetration, synthesis, assembly, maturation, and release. But these particular kinds of viruses, called tempophage, can also alternate between a lytic cycle and an alternative cycle called the lysogenic cycle. In the lysogenic cycle, these tempophage can incorporate the viral DNA into the bacterial chromosome. Notice if we look at the right-hand side image of the lysogenic cycle, when we have essentially penetrated or introduced the viral DNA into the bacterial cell. On the right-hand side, the lower image, where the viral DNA has spliced or integrated into the bacterial chromosome, we now see that we have formed instead what's called a prophage. The prophage is that integrated viral form. Now, every time that DNA, which is now a recombinant DNA, so this is a naturally occurring product that we would define as recombinant DNA, DNA from two different sources. Every time that DNA in the form of this recombinant DNA chromosome undergoes replication or transcription, everything gets copied. Everything that's incorporated gets used. So it could be with this new prophage inserted, the bacterial cell might have now gained some type of new adaptation or new phenotypic characteristic because the prophage genes can be functional. Now, they might not as well, but we're looking at all kinds of possibilities when new DNA is introduced into a chromosome. So the bacterial cell can undergo many cell divisions, with the prophage hitching a ride along every time. Occasionally, there are environmental cues or nutritional cues that cause the prophage to pop out from the chromosome. It becomes unintegrated. And notice we could go right back to the center image where we have the viral DNA as a separate construct and the bacterial chromosome as another separate construct. And now, depending upon the environmental cues, this cell and the virus captured inside could either enter into a lytic cycle, resulting in the formation of new virions for lytic release, or it is possible that the lysogenic cycle could be re-entered or introduced again and the prophage establishes itself again. That's what the tempophage have two possibilities for life cycle, either the lytic or the lysogenic. And prophage is bacterial virus DNA incorporated into bacterial chromosome.